Hi everyone! My name is Noelle, and this is a presentation about my undergraduate thesis project titled Exploring the Role of Lipid Metabolism in Hepatocellular Carcinoma. Hepatocellular carcinoma, or HCC, is a common type of liver cancer largely caused by environmental factors that induce cirrhosis, including hepatitis, alcohol abuse, and obesity. It is common in the US and has a survival rate of only 19% over 3 years because curative treatment options are limited to organ transplantation and resection. These are major procedures that many patients are not eligible to receive. Additional treatment options include radiofrequency ablation and kinase inhibitor drugs, which can mitigate symptoms and extend lifespan but are generally not curative. Due to societal lifestyle changes, the frequency of HCC is expected to increase drastically in the near future, and so it is imperative that we identify new therapeutic options to treat this disease. Liver cells are unique in that they are one of only 3 tissue types in the adult body that is capable of producing fatty acids. It has been found that lipid synthesis and oxidation enzymes are often upregulated in many cases of HCC. The figure on the right, from a study completed by Sanginento and colleagues in 2020, shows 4 critical lipid synthesis enzymes and their regulatory transcription factors whose expression was found to be upregulated in tumors isolated from HCC cells. This demonstrates that HCC could be exploiting lipid metabolism to obtain energy and resources to fuel its development. Therefore, we hypothesize that by inhibiting these pathways, we can selectively slow the progression of HCC by depriving it of the energy and resources it needs to grow. This project seeks to illuminate how HCC cells may be exploiting lipid metabolism to fuel their growth. The metabolic scheme of lipid synthesis and oxidation is shown in the figure, and as you can see, it is very complex with many different enzymes involved. In order to identify possible therapeutic targets within these pathways, we need to complete a broad survey of lipid metabolic enzymes to determine their effect on HCC development. We believe that the most efficient way to complete such a high-throughput survey is through pharmacological inhibition of enzymes involved and quantification of the resulting growth effects. In order to begin this study, I identified lipid metabolic enzymes of interest through an in-depth literature review. I paid special attention to past studies of differential enzyme expression within various carcinoma subtypes. In addition, I included essential enzymes involved in lipid synthesis and oxidation pathways. I then assembled a library of drugs to selectively inhibit these enzymes and screened all of the compounds across 2 different HCC cell lines with different genotypes. The relative proliferation effect produced by each compound was assayed using Alamar blue stain in a plate reader. The strongest hit compounds relative to other compounds were selected for a dose-response analysis. Based on the results of these experiments, we selected a few compounds on which to perform a metabolomic analysis using mass spectrometry. The drug library was purchased as a set from a lab supply company and included a total of 187 compounds. The distribution of the top 15 target enzymes of the compounds is shown on the right. Cytochrome P450 inhibitors are the most highly represented class because these enzymes are involved in the metabolism of nearly every clinical compound, so many drugs inadvertently target this enzyme family. They are also responsible for lipid-based signaling molecule synthesis. The remaining top targets are enzymes more traditionally associated with lipid synthesis and oxidation. The library included compounds with a range of clinical applications, from those already in use for treatment of lipid metabolic disorders such as heart disease, type 2 diabetes, and fatty liver disease, to those only in experimental use that had never been in a person before. 2 doses were prepared from the stock solution at 10 micromolar and 1 micromolar to treat the cells. These graphs are the initial results showing the hit compounds from the overall screen in both cell lines, Hu7 and SNU398. A hit compound was qualified as producing a proliferative effect of greater than 1 standard deviation less than the mean of the 96-well plate. These graphs show the z-score calculated for each hit compound relative to other hits at the high on the x-axis and low on the y-axis dose. Generally, the greatest hit drugs produced an anti-proliferative effect in both of the cell lines, as seen by those in the lower left-hand quadrant of each graph. These were ultimately the compounds we were most focused on for further follow-up studies, including a dose-response analysis. This graph shows the distribution of the enzymes targeted by hit compounds. The top 3 categories, cytochrome P450, autophagy, and phospholipase inhibitors, are representative of the overall distribution of the library. In addition, we believe that they may potentially be cytotoxic because they produced such a negative effect on proliferation. Therefore, we are most focused on the middle range targets for follow-up. This is because in a cancer therapeutic search, we aren't looking for compounds that simply kill all the cells they contact. We are looking for more selective proliferation limiting compounds, which would correspond to those classes with a slightly lower overall effect. Dose-response analyses were performed across 3 HCC cell lines, Hu7, SNU398, and HepG2s. Doses ranged from 0 micromolar to 10 micromolar, and these graphs only include results from compounds that had an IC50 value within this experimental range. The compounds we are most interested in from these graphs are those with a moderate proliferation limiting effect that show up in the middle of each graph. In the Hu7 and SNU398, we can see PF044 and PF3845 as this type of compound. These results were used to inform further follow-up choices for mass spectrometry analysis. As part of a collaboration with Dr. Kim Evison's lab, this library of drugs is also being screened in zebrafish to determine in vivo results of the compounds. The zebrafish have a genetic mutation in their beta-catenin gene that causes liver overgrowth consistent with an HCC phenotype. The degree of liver overgrowth in treated versus untreated fish is used as a proxy to represent HCC progression. The fish are treated as larvae, so they're transparent, and their liver size is easy to observe. Less liver overgrowth relative to the control indicates a hit compound. Currently, only one of the 3 plates of drugs has been completed, but the Venn diagram on the right shows overlap of hit compounds from each of the 3 models used. Intersecting compounds, like 3-nitrocoumarin and PF3845, are of high interest to us because they are proven to be effective in vivo as well as in vitro. Based on the overall screen result data, overlap with fish results, and dose-response analyses, we selected fatty acid amide hydrolase as an enzyme of interest and chose 2 compounds from the library to inhibit this enzyme for mass spectrometry analysis. Both of these compounds are from Pfizer. One of them is preclinical, meaning it's never been in people before, and the other went through phase 2 clinical trials for the treatment of osteoarthritis. It was shown to have anti-inflammatory effects similar to the nonsteroidal anti-inflammatory drug naproxen. Fatty acid amide hydrolase is responsible for the breakdown of anandamide, whose products are used in the synthesis of signaling molecules. It's been found that the buildup of anandamide suppresses the growth, migration, and viability of cells. Endocannabinoids are a class of lipid-based signaling molecules, including those metabolized and produced by fatty acid amide hydrolase. Their role in regulating hunger, sleep, and stress responses has long been appreciated, and more recent studies have found that they are also involved in immunity, inflammation, and cell growth. These pathways are crucial for cancer development, and so they have been highlighted as having potentially oncogenic roles. Fatty acid amide hydrolase is expressed more highly in the liver than in any other tissue, and is primarily responsible for the breakdown of anandamide into arachidonic acid and ethanolamine, whose pathway is shown on the right. These products are used as signaling pathways to promote survival, whereas excess anandamide causes an increase in reactive oxygen species, leading to cell death via oxidative stress. Glutathione is a molecule that neutralizes reactive oxygen species and is often used by cells to combat the effects of this oxidative stress. Shown here is a heat map of relative quantities of metabolites in treated versus untreated cells, which we obtained by running our samples on a mass spectrometer. The first three columns are the control untreated cells, the next three are PF044 treated, and the final three are PF3845 treated. Blue corresponds to higher levels of expression, whereas yellow is lower. The most significant result of this analysis is circled in red, and that is that glutathione levels in treated cells were 59% greater than in control cells. We hypothesized that this is an adaptive response by the cells to compensate for oxidative stress produced by anandamide buildup in the cells due to the inhibition of fatty acid amide hydrolase. In conclusion, the findings of this study don't exactly support my initial hypothesis. I was expecting to see the greatest anti-proliferative response from compounds targeting rate-limiting enzymatic steps in lipid synthesis and oxidation. Though I did find an anti-proliferative effect from compounds inhibiting lipid metabolic pathways, the most efficacious target enzymes were those involved in producing lipid signaling molecules, like the cytochrome P450s and fatty acid amide hydrolase. To me, this demonstrates that HCC cells are exploiting lipid metabolism for their growth, but not because they're using it for energy or resources. Rather, they need to produce signaling molecules and advance cell-cell communication so they are able to grow at an accelerated rate. I also wanted to mention some limitations of my study. First, we need to screen this library in a non-cancer cell line to determine whether our results are cancer cells specific. In addition, my sample size was relatively small. I ran 3 replicates of each dose and compound and averaged the values, but I think a sample size of 20 or so replicates would help cut down on noise and clarify results. Finally, we need the full library to be screened in fish to determine in vivo results. I would like to thank Dr. Greg Ducker for his support, guidance, and mentorship throughout this project. Funding was provided by the Undergraduate Research Opportunity Program. Thank you so much for tuning in. I hope that you found it interesting.